Thank you very much, Vince. I'm really sorry that I messed up the program. It was, I did not understand the passport regulations. But anyway, I'm pleased that I was able to make it. I was supposed to give 2 talks, so I prepared my lecture accordingly. But because of all the changes, I have to now synthesize this somehow to make good sense out of it. What I will do in the first half is to tell you about the clinical manifestations of zinc deficiency and tell you the mechanism, how does zinc work with respect to immune functions, growth and development, and as an antioxidant and anti-inflammatory agent. And in my second part, I will tell you about the clinical impact of the discovery of human zinc deficiency, which has been truly tremendous in the past 20 years, and also discuss the biomarkers of zinc deficiency, which is truly a major issue currently at this time. Just to give you a historical perspective, for the first time that zinc was considered to be essential for growth of a fungus, Aspergillus niger, was reported by Rawlin, who used to be in Lyon, France. And this is interesting because UNESCO started there. Guy Chizot was from Claude Bernard University, and he was a very integral part in development of UNESCO Trace Element Institute. And part of his inspiration came from Rawlin. Actually, if you enter the university, go to the chemistry department, there's a big statue of Professor Rawlin. And also the street is named Rawlin Street. And when I went to Lyon, that was the first thing that Guy Chizot pointed out and showed me. Well, it took many, many years, almost 50 years before it was recognized that all zinc is needed for the growth of higher plant life. And several years later, shown to be essential for the growth of the rats. This is the Wisconsin group of investigators. And finally, in the pigs in 1955. And actually, if you look at the pigs, zinc-deficient pigs, they look just like patients with acrodermatitis enteropathica. There is a great similarity, skin changes, parakeratosis, ulcers, and so on and so forth. And Odell and Savage showed that it is also needed for the poultry. Now, however, so far human were concerned, if you look at the textbook of medicine, biochemistry, or nutrition, they always said that zinc is not needed for growth and development for human. So, but our studies in 1963 showed that this was not the case. Actually, I want to point out here one thing, that this patient that you see here, is a 21-year-old fellow. He was literally, the whole field of zinc started with this patient. He was shown to me in a medical center grand round in Shiraz, Iran. I will tell you later how I got there. But he had tremendous growth retardation, looked like a 10-year-old boy. He had hepatosplenomegaly, his rough skin, mentally lethargic, and he was very anemic. And the reason why they showed me this patient was because he had severe anemia. And it did not take too much effort on my part to determine that it was iron deficiency. Hypochronic macrocytic picture, serum iron was low, iron binding capacity was high. But a strange thing was that he had no blood loss. And if you read the textbook of medicine, hematology, everyone said that there could be no iron deficiency in adult males, unless there is blood loss. So that was one puzzling aspect. And I convinced myself that iron deficiency will not account for growth retardation. So I went to the periodic table, and zinc is in the same transitional element period. And because zinc was shown to be important for the development of fungus and rats and pigs, I hypothesized that we are dealing with zinc deficiency in human. Of course, nobody believed me. It remained a very great controversy, and it took several years later that it was declared as essential element for human by United States Congress, National Academy of Sciences. Now here I have summarized the major manifestations as we described from the Middle East: growth retardation, hypogonadism in males, rough skin, poor appetite, mental lethargy, intercurrent infection. This, I was very surprised that I never saw a dwarf older than 25 in my tours in the villages in Iran and Egypt. And I always wondered as to what happened to them, and it appeared that they were dying because of infection: parasitic infection, viral infection, bacterial infection, pneumonia, meningitis. And that gave me the idea that most likely zinc was needed for immune functions. And when I came to United States, I devoted most of my time in trying to understand how does zinc affect immune cells. Now, as I told you that initially in textbooks, zinc was not important and zinc deficiency did not occur. Today, it's the World Health Organization estimate that zinc deficiency is common throughout all the countries, mostly in the developing world. The reason is that they all subsist on cereal proteins. And now we understand that cereal proteins has an organic phosphate compound, phytate, and that complex is zinc. And so zinc is unavailable for absorption, and there is zinc deficiency. For instance, look in the Southeast Asia, something like over 90% of the villagers, they are susceptible to zinc deficiency. Now, what about United States, Canada, and developed countries, Europe, and so on? Well, zinc deficiency is also present, but in certain populations. Like we have studied the elderly in Detroit area, Michigan. Prior to 1990 or so, everyone said that their plasma zinc is normal, they are not zinc deficient, but we showed that indeed they were zinc deficient, their lymphocyte zinc was low, their immunological effects were affected adversely related to zinc, and they had oxidative stress and also generation of inflammatory cytokines. And all these were corrected by zinc supplementation. And our estimate is that something like 30 to 50% of the elderly living in United States, and they are well-to-do, they are eating well, they are ambulatory, they have no diseases, but they are zinc deficient. Question is, why are they zinc deficient? Well, first is that, as you know, there's a great tendency now to move on to cereal protein, and cereal protein again has unfavorable phytate content for zinc. So we have that problem, and I'll show you some data to show that indeed that is the case in Michigan elderly population. So what about women who are pregnancy and all? You see, if you look at the RDA, recommended dietary allowance, the intake should be around 25. Now, if you have a very good diet, well balanced, you have animal protein and you have fruits, vegetables, milk, and everything, but your caloric content has to be around 2200. And so normally, it's very hard to reach more than 12 to 13 milligram of zinc intake under the best of circumstances. How are you going to get 25, 20 for women who are pregnant and who have pregnancies and feeding children? It's not possible. Physicians have to consider supplementation. And this was not the case, but apparently now things are moving in the right direction. And what about the population, such as American blacks and Spanish population and so on? There has been several papers published that something like 25% of these population also are susceptible to zinc deficiency. So the point I would like to make here that we have in our certain population, they are very susceptible to zinc deficiency, and the physicians must understand this problem. Now, then there is something else that has happened since then. If you walk around in the hospital wards and look at the patients who have chronic renal disease, we published many papers in the 80s that the people who have chronic renal disease, they have zinc deficiency. How about chronic alcoholism? It is very well known that you excrete a lot of zinc in the urine. Alcoholics, they become deficient. Cirrhotics, they are known to be zinc deficient. Malabsorption syndrome, and many of the cancer patients. So, but do you realize that, most of the time, this is not diagnosed properly in the hospital. Physicians don't think of it. And at least if you think of it, that zinc deficiency is a problem in some of your chronically ill patients, you can at least do a few things. Correct the deficiency, their immunity will improve. Many of them die because of repeated chronic infections, repeated bacterial pneumonia, meningitis, and the incidence will go down. I will tell you a little later how it happens. So, infection, immunity, healing, and also cognitive impairment. Some of these things are affected because of zinc deficiency in the chronic diseases. Now, little bit about the advances in the basic science area. When I started my research in early 60s, we knew of only 3 enzymes that were zinc dependent. One was carbonic anhydrase, other was carboxypeptidase, alcohol dehydrogenase. Now, none of these would account for growth and development, immunity, and so on and so forth. So the question was that was zinc important so far the enzymes were concerned? Now we know that close to 1,000 enzymes, they require zinc either for their stability of a structure or for activity. However, the dogma was in the early 60s that no matter how much deficiency there may be in the human, enzymes will not be affected because of zinc deficiency. The reason was that so little zinc was needed for these enzymes that the body will not give it up. And so they will hang on to zinc, enzyme function will remain intact, and there will be no effect of zinc deficiency on the enzymes. This was the same argument also given for the many of the transcription factors. Now we know that there are 2,000 transcription factors that require zinc for their stability and their role in gene expression of many proteins. And the same argument was that no matter how deficient the organism is, human, there will be no effect on these. But our first effort was to show what happens to enzymes, and then later on we showed about the transcription factors. We showed that many enzymes were affected, and critically there were 2 enzymes, both in human and cell culture model, which were very critical. One was the deoxythymidine kinase, an enzyme needed for the incorporation of thymidine as TTP into DNA and for cell division and therefore growth. And this turns out to be a zinc-dependent enzyme. There is no zinc in the molecule, but our study showed that zinc was required for the gene expression of deoxythymidine kinase. Now, this study was very difficult to do in human because you need a proliferating tissue. And what we did was to take the zinc-deficient subjects, implanted a sponge under the skin, harvested the collagen in a week or 2 weeks, and we did an acute experiment and showed that indeed deoxythymidine kinase was affected adversely in human. Now, and the other enzyme was the 5' ectonucleotidase in the lymphocyte. And this work was done by my daughter, Sheila Mehta, who is in the audience. This was her PhD work. And they showed that this enzyme was also very sensitive to zinc restriction. Not only that, but if you measure this enzyme, you will find that it is affected adversely within 8 to 10 weeks of the restriction of zinc. However, at that point, if you measure plasma zinc, it will be completely normal. And if you measure lymphocyte zinc, even that will be normal, indicating that some of the other parameters are more sensitive as a biomarker than measurement of zinc in the plasma, urine, hair, red cells, lymphocytes, and so on and so forth. I'll come back to that in a minute. And now we know that the body has a very intricate mechanism to maintain intracellular zinc level. For instance, there are 14 ZIP and 10 ZNT transporters. The function of the ZIP is to bring the zinc from outside to inside the cell, and the function of ZNT is to take the zinc out from the cellular compartment in order to maintain a physiological level of intracellular zinc inside the cell. And it is turning out to be a very exciting area. As a matter of fact, the fact that zinc is now recognized as a molecular signal, just about 10 years ago, a Society of Intracellular Zinc Biology has been established. And there are 150 molecular biologists working on this aspect of zinc as a molecular signal. And how does zinc work in ZIP and ZNT transporters? And how does zinc signal the cells to perform their duties? Now, I'll just tell you very briefly, how does zinc affect the growth? Well, we did a lot of studies with growth hormone and many other. It turns out that IGF-1 is zinc dependent. If you restrict zinc in human, IGF-1 declines. And so it appears that zinc is needed for the gene expression of IGF-1. And IGF requires a receptor when it goes to the cell. And this receptor has tyrosine kinase. And tyrosine kinase must be phosphorylated. And zinc is required for the phosphorylation of tyrosine kinase. And then IGF will go inside the cell, and it will then upregulate the uptake of thymidine inside the cell for DNA synthesis and cell division. So that's what we think is the mechanism. And the enzyme there used by the cell is deoxythymidine kinase. So we think that the effect of zinc on growth is number 1 on IGF, number 2 on IGF receptor tyrosine kinase, number 3, it is needed for the conversion of DTMP to TTP by deoxythymidine kinase. And we believe that this is the mechanism of zinc on cell growth. Here I have summarized these that I have already explained to you. Now, it also turns out that the deficiency of zinc, we described from the Middle East, there were adolescents, there were ages of 16 to 21, those are the ones that we studied in the Middle East. and they were more or less a moderate type of zinc deficiency. They were living, but their lifespan was not normal. They will die by the time they were 25. But there is another variety of zinc deficiency, and that's a very severe zinc deficiency seen in patients with acrodermatitis enteropathica. They have tremendous immunological problem, cell-mediated immune dysfunction, loss of hair, and if you look at these around the eyes, parakeratosis, around mouth, and they died usually with this disease no more than 16 or 18 years of age. So it turns out that this acrodermatitis enteropathica is due to a genetic mutation of ZIP4. This is a transporter which is needed for in the intestine to take the zinc from the intestine to inside the cell. And if it is mutated, they do not absorb zinc. And for the first time, it was reported in only a short letter to the editor, 10 sentences by Professor Moynihan, that you can save the patients with acrodermatitis enteropathica by giving zinc. And now it has become a standard treatment. And of course, now we know that it's a mutation of ZIP4. It's not a very common disorder, but it's prevalent all over the world. Now, this one is not acrodermatitis enteropathica case. This was a patient in Morgantown with Wilson's disease, and he was receiving penicillamine for treatment of Wilson's. And they gave too much of penicillamine and became very zinc deficient. And Professor Klingberg called me from Morgantown, and he sent me samples of blood, hair, urine, so on. And we determined that indeed he was very zinc deficient. And I recommended that he treat him with zinc and stop penicillamine. And after that, he recovered completely. And he was going to college at this point. So the point here is that you have severe deficiency of zinc. Manifestations are very severe. Must recognize and treat. You don't treat these patients with 15, 20 milligram of zinc a day, but you have to give 75, 100, rather therapeutic level of zinc in order to overcome the deficiency. Now, what about the mild deficiency of zinc? This was my major effort, because if you look at the spectrum, you have severe deficiency, moderate deficiency. There must be a spectrum of mild deficiency existing in any given population. And our approach was to have an experimental model of zinc deficiency in volunteers, adult volunteers. We restricted zinc 3 to 5 milligram a day. It was a very difficult to study, very complicated. We had to use semi-synthetic diet, experimentally produced, and we were very successful. We were supported by NIH and FDA. Initially, I had a hard time getting permission to produce zinc deficiency in human, but I was permitted by all the agencies. And we found that indeed, even if the deficiency is very mild, there are many manifestations. Most importantly, I found that the natural killer cell activity was decreased. There was a decrease in the production of interleukin-2, and there was a decrease in the activity of thymolin. And thymolin, as you know, is a thymic hormone, very critical for the differentiation of T helper cells and proliferation. So it was just around that time that Professor Jean-Francois Bach from France reported that thymolin is a non-apoptide and zinc is sitting at the active site. So I got very excited about the possibility of looking this into my experimental model. I made a trip to Paris, had a conference with him and Nardin, and we did many studies in human about thymolin. I'll tell you a little more about that. And then also Dr. Sandstedt showed in the Chinese study that impaired cognitive functions were related to mild deficiency of zinc. Now here I just have a cartoon to show you how is zinc working in the immune cell. Well, if you look on the top left-hand side, that's where thymolin is, and this is zinc dependent, and thymolin activity is decreased. And one of the functions of thymolin is the differentiation of T helper cells in Th1 and Th2, and also the proliferation. So, and what is Th1? Th1 is the most critical helper cell, which is responsible for generation of interleukin-2 and interferon gamma. Interleukin-2 is needed for the activation of natural killer cells and T cytolytic cells. And these are the cells that kill the tumor cells, the viruses, the parasites, and some unusual bacteria. So it's very critical to have a good function of natural killer cells and T cytolytic cells. If interleukin-2 is decreased, these functions are affected adversely. And the other very important cytokine is interferon gamma. And interferon gamma, along with interleukin-12, which is generated by the macrophages, monocytes, is a very powerful tool for the monocytes, macrophages to kill the parasites. It's one of the most powerful tools that we have at this time. So, thus, if the differentiation of T helper 1 is affected adversely, you will have problem with cell-mediated immunity. Now, we did not find any effect on T helper 2 function. So, interleukin-6, interleukin-4, interleukin-10, they were actually interleukin-10 was affected. As in our studies, we found that interleukin-10 was increased, and this has a negative effect on Th1. So this further decreased the generation of interleukin-2. But most importantly, and this was a great surprise for me, that here we had experimental model of human zinc deficient subjects. They had no infection. They were perfectly stable. All they had is mild deficiency of zinc. But when we measured interleukin-1 beta, we found that that was increased. And that gave me the idea that zinc deficiency per se is a stress factor for the monocytes and macrophages. And we went after that, we looked into the cell culture model and human studies, and we found that indeed, with zinc deficiency, the TNF-alpha production, interleukin-1 beta, interleukin-6, interleukin-8, these are all increased. So it tells me that zinc supplementation is very critical as an anti-inflammatory agent. You must have proper zinc in order to control the generation of these inflammatory cytokines. And the next question comes up, what causes the stress to these monocyte macrophages as a result of zinc deficiency? And I will have a diagram to show you later how this oxidative stress is caused by zinc deficiency. Now, one other thing I'd like to point out here is that this led me to look into a lot of transcription factors. And we have now shown that we have shown at least 6 transcription factors that are affected adversely because of zinc deficiency. 3 of them are involved in the interleukin-2 production, like NF-kappa B, AP1, SP1. There are TBET, STAT4, and interferon gamma. They are involved in the differentiation from Th0 to Th1. They are also affected adversely. And we show that A20, another transcription factor which is generated by monocytes, macrophages, they are adversely affected if there is zinc deficiency. This is just to show you a scheme of binding of interleukin-2 to the promoter area of the gene. And as I mentioned, that we found that NF-kappa B, nuclear factor kappa B, and AP1, SP1, they are very critical. They must bind to the IL-2 gene promoter area in order to generate interleukin-2. And here I'm showing you very briefly what happens to NF-kappa B in T helper 1 cells. If there is zinc deficiency, NF-kappa B generation is decreased, AP1 is decreased, and SP1 is decreased. So there's a very critical effect of zinc on T helper 1 cells on these various transcription factors, which affects the generation of interleukin-2, and then the effect on NK cells and T cytolytic cells. Now, this is a picture that I'm showing here in D. If you look at the panel D, this is where I'm showing that NF-kappa B, once it gets activated and zinc must be present, then it will go to the nucleus and then it will bind to the IL-2 promoter. Now, if there is zinc deficiency, like in other panels, there is, like C, is NF-kappa B will not go to the nucleus and will not bind to the IL-2 promoter. Here I'm very briefly showing you the role of zinc. And this is, I must emphasize to you that the effect of zinc on NF-kappa B activation is cell-specific. This is, I'm showing you only for the T helper cells. Now, zinc is required for the generation, for the gene expression of NF-kappa B. And the arrow goes there on the P105. And this has two components, P60 and P65 and P50. And what happens to NF-kappa B, that in the cytoplasm, it is bound to an inhibitory protein, I-kappa B. And this keeps NF-kappa B bound together. Now, this must be released. I-kappa B must be released for NF-kappa B to be free and then go to the nucleus and activate interleukin-2. And what zinc does, it phosphorylates an enzyme IKK alpha, and that will phosphorylate I-kappa B and split I-kappa B from NF-kappa B. And that's what I'm showing you here in this cartoon here. And by this way, NF-kappa B is activated by zinc. And NF-kappa B activation is needed not only for interleukin-2, but also in interleukin-2 receptor alpha, interferon gamma, and generation of NF-kappa B itself, and I-kappa B alpha, the enzyme that phosphorylates I-kappa B. So it's a very critical step in cell-mediated immune function. Now here I'm showing you very briefly on Tibet messenger RNA. If there is zinc there, it is adequate. If it is zinc sufficient cell, 1 macromolar, it is very decreased. And here I have a picture of the Tibet in a STAT4. But the basic point I want to make, that the dogma that zinc deficiency will not affect transcription factor is not correct. Our research has shown that at least in 6 transcription factors, we have shown that it is affected adversely as a result of zinc deficiency. So what about many other transcription factors involved in zinc signal? Well, this is for the whole host of molecular biologists that they have to work on it and define further effect of zinc on molecular signal. Now, here I'm trying to show you how does zinc affect the oxidative stress. As you know that the oxygen, it loses 1 electron, it becomes superoxide free radical. And one enzyme that does that in monocytes macrophage is the NADPH oxidase. So NADPH oxidase is activated, you have more free radicals. Turns out that zinc is an inhibitor of NADPH oxidase. So this will then decrease the generation of superoxide. And the next step is that once superoxide is being generated, it converts to a hydrogen peroxide, and the enzyme is superoxide dismutase, which is both a zinc and copper enzyme. and zinc is essential at that step. And then at the next step, where hydroxyl ions are being generated, these are also equally very bad molecules for oxidative stress. And there are two mechanisms. One is that if there is iron and copper, they will accelerate generation of hydroxyl ion. And zinc will compete with iron and copper. So that's another positive effect of zinc on generation of hydroxyl radical. And then zinc is involved in generation of another protein, metallothionein. Metallothionein is zinc dependent, and this is an excellent molecule to sop up all the hydroxyl radical. So basically, I'm showing you here several mechanisms why zinc is antioxidant. Now, why is it important to know? Because if you look at the current literature about chronic diseases, such as atherosclerosis, some cancers like prostate cancer, Parkinson's disease, many other neurodegenerative disorders, everywhere people think that it's the oxidative stress and chronic inflammatory cytokines that are the culprit. They are responsible. For instance, these days, cardiologists, they always measure CRP in patients with atherosclerosis because they think there is an inflammatory cytokine involved. So zinc is a very powerful anti-inflammatory substance and antioxidant. So therefore, I think that perhaps more studies in future will show that zinc may have a very important preventive effect in atherosclerosis and some cancer. I'll come back to this a little later I think I will go back to, here I have a list of a lot of collaborators. This is a common list. You will see it in the next presentation also. Could I have the next, my PowerPoint? I will move on to that. Is somebody to change the PowerPoint for me?
While they're doing that, I just want to let the audience know that 3 of the presentations that are scheduled have been canceled, so there's plenty of time going forward. Specifically, the talk that is scheduled for 11:30, 12:20, and 13:10, those speakers are not here. So after Dr. Prasad finishes his second, then we'll break for coffee.
Well, wherever I go to present my talk, people think that I'm Iranian, and I discovered this in Iran. But I was born in India, and how I got there to Iran is a very interesting story. I was very lucky to be accepted in the Department of Medicine under Cecil Watson. He had a program after World War II where he was training clinicians as scientists. It was a clinical scientist program. And the idea was that they will not only be a very good clinicians, understand medicine, but they will also take the problems on the bench side and solve it with the proper biochemical studies and physiological studies. And there were very few universities who had started doing it after 1945. Cecil Watson was one of them. And he used to take about 2 or 3 students every year like that. And I was exceedingly fortunate to be accepted by him in this program. And that made me a clinical scientist. I got a PhD. But it gave me so much of knowledge and tool in the basic science that I could take the problem in the laboratory and solve it. Usually the case was that PhD was working with the basic science. They did not know anything about the medicine, and physicians were working with the patients. They did not know anything about the basic science. And people like us as clinical scientists they were filling the gap. They were bridging the gap between the two. So this prepared me. And then what happened that after I finished my training in Minnesota, Hobart Ryman, who was chief of medicine at Jefferson, he was a personal friend of Shah of Iran, and he invited him to set up a medical curriculum like an American school, and he was going to Shiraz. And he contacted me if I will join him. And my first reaction was that I don't want to go there. I'm not an Iranian. I don't know the language. I have no interest. And my work in Minnesota was going very well. So why should I leave? But he convinced me that 2 years of duty in a foreign country will be good for you. It will enlarge your vision in medicine. And even if you can't do a laboratory research, at least you will have another perspective in your career. Well, I accepted the challenge, went there, and it so happened that his words were so true. I saw that patient that I presented to you earlier in Grand Round, and that opened up the whole area of zinc metabolism in human as you see it today. It was completely unknown prior to that. Well, when I first proposed that it might be zinc deficiency, it was taken as a great skepticism throughout the world. All the scientists thought that I was wrong, I was missing some other element or some other amino acids and so on. It can't be zinc deficiency. Well, I wanted to get some zinc measurement done in Shiraz. I had 12 patients. I studied them in great detail, but no zinc measurement. And I contacted Bert Valley in Boston. He was the only one in the whole world who had a setup with diathiazide technique to measure plasma zinc. And originally he accepted to help me, but I never got any results back. I sent him all the samples. So I was, meanwhile, anxious to leave Shiraz and come back to the States or somewhere else. So I decided to publish this paper as a syndrome of iron deficiency anemia, hepatosplenomegaly, hypogonadism, dwarfism, and geophagia. Interesting part about the Iranian dwarfs was that they were eating also clay. A pound of clay every day. I've never seen a clay eater in Minnesota. So it was a very strange situation. But I presume that because of high phosphate content in the cereal protein and the clay, they might not be absorbing iron and zinc. So they had a syndrome combining the two elements. And that was the whole thesis of this paper. The editor was Alexander Gutman, a very brave editor. He did not get it reviewed by anyone else but himself. And I told him that I'm speculating about zinc, and I will be able to remove that part if he didn't like it. But he said that he likes that the most, and he never removed even one sentence. Everything that I wrote was published in a 16-page long pages of Green Journal as a syndrome. And this attracted the attention of a lot of professors of medicine and nutrition. One of them was Professor William Darby at Vanderbilt, and he invited me to meet him in Egypt because FDR had established the United States Naval Medical Research Unit. The purpose was to study infectious diseases, and actually the entire history of typhus fever was written there in that institute by Rockefeller Group. And they had just finished their work there, and the institute was available for other studies. And Bill Darby negotiated with the Navy to give this for studying zinc metabolism. Well, my problem was that I thought that if I went to Egypt, and what if I never saw these patients? So what happens? Because my experience was only in Iran. So I told Bill Darby that I would like to go to the villages and look for them. And here is one patient in Egypt. The other one is a physician, Iranian, Egyptian physician. Thank you very much, David. It helps me. And this dwarf, he was 20 years of age, but looked like an 8-year-old boy. I saw many of them in my tour in the villages in Egypt, and I accepted the challenge. I told Bill that, yes, I will come to Egypt and investigate this problem. And I got $90,000 grant from NIH to do this work, and the Navy supported, and Vanderbilt gave their biochemist to work with me, and so on and so forth. So my team was established, and I moved there. The parting message from Bill Darby was that, Ananda, I know that at the end of 2 years you will be proven wrong, but it should not discourage you. You are a very hardworking, intelligent individual. You'll probably move on to some other areas of good research. So that was the parting message. Well, it turned out that I was right. I first established dithiazone method. It was a very difficult method. I used to work in the lab 8 hours a day and get 2 results at the end of the day. And we studied 40 dwarfs. We found that plasma zinc was low, red cell zinc was low, urinary zinc was decreased, and we were exceedingly lucky to get permission to use zinc 65 tracer dose to study 24-hour exchangeable pool, turnover rate, and excretion. And we published all these results in this paper, a Journal of Laboratory and Clinical Medicine. And this paper has been republished as a landmark article. And this is the paper that has been picked up by American Physiological Society and American College of Physicians as one of the most important articles published in their field in 100 years. So, and after this publication, we started supplementing them with zinc. And turned out that once we gave them zinc, they grew at a rate of 5 to 6 inches in height in first year. Their gonadal changes took place within 3 months and remained anemic. Whereas those who received iron, they corrected anemia. Nothing happened to their growth and gonadal changes. So basically, our studies showed very convincingly that zinc deficiency occurred and was very critical for human development. Now the controversy begins after that. Everyone, for 10 years, there was argument in many journals, including Lancet, that zinc is some editors said that zinc was very important, great discovery, but all others said that it was all problematic, it could not happen. And one of my friends called me from New York, said that you are the greatest object of controversy in Lancet. You should go to the library and see. So I went there, and every issue had pro and con, pro and con. I got into the act, and I also wrote an article. And finally, Moynihan's paper appeared in '73 showing that acrodermatitis enteropathica was a genetic disorder, and it was correctable by zinc supplementation. And next year, I got a call from National Academy of Sciences to meet with them because they were considering zinc as an essential element. And I was very happy to get that call. I went to Washington, and US Congress declared zinc as an essential element. And all my controversies vanished, and my life became very simple and easy. Now, here I have listed some of the impact which has happened since in zinc area clinically. Well, as you know that in Bangladesh, in India, in Pakistan, and many other developing countries, acute infantile diarrhea has a very high mortality rate. Almost 90% of these children die within 3 weeks. And they were giving usually the same solution which was designed for cholera treatment for rehydration. I was invited by UNICEF to go to Bangladesh. I looked at it and I told them that why don't you include zinc? It's an essential element you were missing. And they agreed. We decided on 15 milligram of zinc supplementation. And immediately they found that the mortality had decreased to around 15–20%. And this was repeated in Pakistan, India, and all over the world. And today, WHO has made it mandatory to use zinc for treatment of acute diarrhea in over 70 countries. And Bill Gates Foundation gave $20 million for doing this work in 20 different countries in Asia and Africa. And this thing is truly has decreased the mortality in a very, very large number of infants and children. Now, the problem is, however, that they are being treated acutely. They live, their diarrhea has been cured, but then they go back to the villages and they remain deficient. And what will happen to them? Well, then they will happen to those dwarfs that I saw in Iran and Egypt. And eventually they will die at the age of 25 because of cell-mediated immune dysfunction. So this was my argument with Gates Foundation 2 years ago, that you are doing a great job, you give $20 million, saving the lives, but you are creating a whole bunch of zinc-deficient subjects in the developing countries who will die at the age of 25. Do something about that. And I was able to convince them, but in the past 2 years, all the hierarchy of Gates Foundation, I can't describe to you how difficult it is. The whole scenery changed. I was dealing with Dr. Brewer. He left, Tom Brewer, not George, Tom Brewer. And he left, and now we have a new group of people. And now I'm dealing with the new people, and the whole thing has been postponed and delayed. Second thing, Wilson's disease. It turns out that zinc is a very important therapeutic modality for treatment of this fatal disorder. And my good friend George Brewer in Michigan, he has been working on zinc as a treatment for Wilson's for the last 20 years. And it has now been approved by FDA as a therapeutic modality. And Dr. Brewer has published many papers. Common cold. It is still, people don't use the zinc properly. If you use zinc, the proper salt, the salt has to be something which releases zinc. Usually what happens that many preparations will have zinc gluconate, but they will add citrate, they will add tartrate to improve the taste. And that will prevent the release of zinc from the salt. And therefore, it's not available for fighting the viruses. And it has to be started within 24 hours of the onset of common cold. So if you did that, within 24 hours, use the proper chemical and proper dose. The dose is that every 3 to 4 hours you have to take a zinc acetate lozenge. Then you will cut down the severity and duration of common cold by 50%. Now, this has been reviewed by Cochrane Review about 2 years ago, a very long article which also appeared in Journal of American Medical Association. So if it is done right, zinc is very effective. Sickle cell disease, the same story. They are very zinc deficient. They have a lot of hyperzincuria. They have growth retardation, immune dysfunction, incidence of common infection, pneumonia, meningitis, and most of the death is because of infection and pain crisis. And again, Cochrane review, recent one, has said that the zinc is the only modality which will decrease the incidence of infection and pain crisis. Now, prevention of blindness due to age-related macular degeneration. As you know, that worldwide ophthalmologists are using preparation of vitamins with zinc, and two things have happened. It has decreased significantly in 27% of the patients with macular degeneration, preventing blindness in them and also activation of AMD. Now, it turns out that if you separate the effect of vitamins with zinc, zinc has most effect. Not only that, but the zinc supplemented patients also had decreased mortality, not the vitamin supplemented. So there is a very distinct effect of zinc compared to the vitamins. And I should mention here about acizole, which is a common treatment for treatment of carbon monoxide poisoning in the Russian study. Professor Anatoly is here from Russia. He knows a lot about this. And decreased incidence of infections in the elderly. Well, our studies in the elderly have shown that not only we improve cell-mediated immunity, but also we decrease chronic inflammatory cytokines and decrease oxidative stress. This is very, look at the first level. I show here that the duration of common cold is almost half. Normally it lasts about 8 days, it's now 4 days if you use zinc acetate lozenges. And here the severity also, by the end of 4 days, the severity is significantly reduced, less than 50%. How does it do that? What is the role of zinc? Well, I think what is happening that there's an endothelial cell adhesion molecule, and this ICAM1 is the cell surface where viruses, rhinoviruses particularly, attach. So if you decrease this by zinc supplementation, rhinoviruses do not have a receptor where they will attach and reproduce and produce and have cold infection. So in effect, I think zinc is working like an antiviral agent by this mechanism. Now here, very briefly, just to show you in the elderly, the incidence of infection is decreased by 66% in our studies in Michigan. As I told you that prior to 1994, 95, zinc deficiency did not occur in elderly. We showed that indeed it was there, and by supplementing you decrease the incidence of infection. Here I'm showing you that the CRP level is also decreased, and this is a marker of chronic inflammation, and that is decreased in zinc supplemented subjects. And this is the oxidative stress, plasma oxidative stress marker, MDA and HAE. It's also significantly decreased. So, and here I'm showing you the effect on generation of interleukin-2 messenger RNA. It is indeed low in those who are not receiving zinc as compared to those who receive zinc supplementation. So basically, in our studies, we have done 2 or 3 supplementation studies in the elderly. We have shown that the incidence of infection is down, chronic inflammatory cytokines are decreased, oxidative stress markers are decreased, showing that these effects are very critical in chronic diseases. Now, this one is my hypothesis slide as to what is zinc doing. Well, first effect of zinc is on the oxidative stress. I already given you the mechanisms. So, and if the oxidative stress is decreased, then oxidized LDL will be decreased. If it's zinc deficiency, oxidized LDL goes up, and what this does, that it activates NF-kappa B inducing kinase and activates NF-kappa B. Now, NF-kappa B in monocytes macrophages is a major culprit. It's not a beneficial agent like what I showed you in T helper cells. So NF-kappa B activation in monocytes macrophages does number of unfavorable things for human health. For instance, one thing it does is activates anti-apoptotic gene. So if somebody has chronic prostatitis, chronic inflammation of the prostate, or prostate cancer, the cells will not die because anti-apoptotic gene is activated. And it also activates growth factors. So the cancer cells will multiply. So cancer cells are multiplying and not dying because of NF-kappa B activation. What about inflammation? All the inflammation like TNF-alpha, interleukin-1 beta, and all gene expression, they require NF-kappa B activation. So, and then we have the adhesion molecules. That's all I'm showing at the bottom. Now, if you can decrease NF-kappa B activation, you will be able to improve all these features. And actually what happens that by giving zinc, monocytes and macrophages, they generate a transcription factor 820 that I'm showing you on the right-hand side. And this 820 via TRAF pathway, it inhibits the NF-kappa B inducing kinase, and therefore NF-kappa B activation is decreased. So it has a very beneficial effect. If we give zinc, you will be able to decrease the anti-apoptotic gene, decrease the growth factor for the cancer cells, and decrease inflammation, decrease adhesive cytokines, and you will have beneficial effect on atherosclerosis. So my hypothesis is that there should be a big trial, perhaps lasting for many years, in a large number of subjects, and see if we can indeed use zinc as a preventive agent for atherosclerosis and perhaps prostate cancer. These are the two that I select from this thing that I'm showing you here as a possible agent. Of course, it could apply to some other neurodegenerative disorders, but I have not studied and I do not know the answer. Okay, now my last section is biomarkers. What do we have? Well, in 1963, when I did all these studies, I did a very hard way. That is the assessment of zinc by dithiazone technique. Very difficult. Nobody could do it. Ours was the second lab in the world that set up this technique in Egypt, dithiazone technique. I came back from Egypt in 1963, and I got a call very soon after my arrival from this person who was developing atomic absorption spectrophotometer. And he called me and he said that we have developed atomic absorption spectrophotometer, commercial variety, and we would like to give you free $50,000 instrument, and your life will be much simpler. You will be able to assay many samples at once. So it was Walter Slevin. He's the one who called me. And the instrument arrived. I was very happy, but I could not measure zinc in the plasma. It plugged up all my tubes and everything. So I called Walter Slevin. I said, your Perkin-Elmer instrument is great, but it doesn't do the job. So he said, oh, Dr. Prasad, I sent you this instrument to set up the method as to how to measure zinc in the plasma and the cells. I said, oh, so I had a 2 year of job to do with the instrument. Well, I finally succeeded in that. Well, and published that method in '65, and then I showed that indeed atomic absorption was useful for measurement of plasma, zinc, and lymphocyte and red cell. Well, before that, I will just very briefly tell you that zinc 65 clearance was increased in the dwarfs as compared to the controls in Egypt. And turnover rate was, like here is the normal 1, it was 1.5. So turnover rate was high, clearance was very rapid from the plasma, and exchangeable pool was very low. So those were the results that we published in our first paper. Now, in this paper, I described the atomic absorption spectrophotometer technique, and you won't believe it that I published in '65. This is the method in 2015 people are using for measurement of plasma zinc by atomic absorption. The problem is with that it's a big instrument, costs $50,000, and it's not available in many developing countries. And my experience in India was that some laboratories had this instrument, but they did not know how to use it. And some people who used it, it went bad and they couldn't repair it. So it was all sitting there in the laboratory. So we are now looking for another way to make it simpler, and I'll tell you what our approach is going to be. This is the paper we published, our experimental deficiency of zinc in Annals, reference. And this is the 5' ectonucleotides and lymphocytes. And here, this is the work of Sheila. And you can see that in those who were zinc supplemented, their enzyme went up very significantly, indicating that the lymphocyte maturity markers were high. It is an enzyme which signifies maturity of lymphocytes. But while this is an early effect in ectonucleotides, if you look at the lymphocyte zinc, it was not affected at that point, and plasma zinc was also not affected. So it tells me that perhaps the measurement of ectonucleotides is much more sensitive marker than the measurement of zinc in the plasma lymphocytes and the cells. So the immune functions are truly very critical. Now, this is about the thymolene. It's the same story. The thymolene activity is affected within 10 weeks of zinc restriction. And this was done by a bioassay. And we hope that we can develop some simpler techniques that will be available to all the laboratories. And here I'm showing you the thymolene activity before and after in patients with sickle cell disease and also in non-sickle cell patients. And very interesting part about thymolene is that if you add zinc in vitro, then you correct the thymolene activity. So you have an in vitro test. You measure the plasma activity of thymolene, add zinc, and you correct it, then you have a diagnosis of zinc deficiency. Whereas if they are zinc sufficient, nothing will happen to the thymolene activity. And same thing is true about interleukin-2, messenger RNA. This is what I'm showing you here. Very early effect of IL-2 mRNA of zinc deficiency. Now, what we did, we looked at the interleukin-2 mRNA in the elderly subjects, and we tried to look into the in vitro correction of IL-2 mRNA in the elderly. And it is true that if you measure the messenger RNA of interleukin-2, you can correct it in vitro. So in my opinion, this could be a very interesting and important test for making a diagnosis of zinc deficiency in human subjects. If you add any other metal like manganese or copper or iron, nothing happens to the messenger RNA, but zinc is the only one that will correct interleukin-2 messenger RNA. There is one other very interesting test that goes on in human deficiency. That is the measurement of absorption of zinc and endogenous secretion of zinc. But for this, we used zinc 70 to do our studies. Here I'm showing you that in depletion, there is a negative test, and when you restrict zinc, there is a, and the absorption is increased initially, but if you continue depletion for 6 months, it's not sustained. Contrary to the idea that because somebody is deficient chronically, he will keep on absorbing more zinc. That's not true. After 3 months, the adaptation is gone. But endogenous secretion keeps on decreasing. So in my opinion, this is also a very good biomarker for zinc deficiency in human. Measure the endogenous secretion with oral zinc 70. Now this is our latest effort. It's being done in collaboration with Chris Fredrickson of Neurobiotechs in Houston. You see, the idea came to us that when Mars probe went, the robot, they used a laser beam on the rocks, and they were able to see beautiful spectrum for zinc. So it occurred to us that perhaps if we can develop a laser technique to measure the background for zinc, we should be able to measure zinc right here in a very simple way. Our concept is that this could be contained in a small box like you see here, and you can carry it in your suitcase and go to India or Philippines or anywhere and use this for measurement of zinc. Now, look at this. You see, it's a sensitivity of laser-induced background spectroscopy is tremendous. For atomic absorption, it's the nanogram. Here we can measure femtogram. So if that is true, then we should be able to isolate thymolene from the plasma with an antibody and measure zinc in the thymolene. At this time, there is no way you can measure that a small amount of zinc. But with this technique, I'm hoping that we will succeed in measuring active thymolene. And this is the result we did in the elderly subjects on the nail zinc. And you can see here that it correlates very well with the phytate zinc molar ratio. If the phytate molar zinc ratio was greater than 15, their nail zinc was less than 140. Those who were normal, they had a higher level of nail zinc and lower molar ratio of phytate to zinc. Now, there's a statistical correlation. Numbers are very small. We had only 28 altogether. But if you look at this number, this tells me that almost 50% of our well-to-do elderly in Michigan had chronic deficiency of zinc, and they had higher phytate to zinc molar ratio. I'm showing you the same data over here. So in summary, what are the biomarkers available? Right now, all the laboratories are only measuring plasma zinc. Is that a good indicator? No. Plasma zinc has a lot of problems. If there is slight hemolysis, it goes up. If there is a stress, if someone is walking and jogging, the plasma zinc is changed. If there is infection, plasma zinc is altered. If there is myocardial infarction, plasma zinc is affected. So it's not a very sensitive, good indicator. But that's the only thing that laboratories have at this point. What do I suggest? Well, If I have my choice and if I can succeed, I will measure serum thymidine activity. Hopefully with laser-induced background spectroscopy, we can do that. I would like to measure the generated interleukin-2 messenger RNA. It could be a very sensitive, specific test for biomarker of zinc deficiency. Lymphocyte ecto-5' nucleotidase is another good marker, but this enzyme, it needs column chromatography. It's not a very simple test. Similarly, deoxythymidine kinase is a very complicated assay. You need a generating tissue to measure that. What about zinc in lymphocytes and granulocytes? Yes, it's a better indicator than plasma, but it's not as sensitive as thymidine activity or interleukin-2 messenger RNA. Intestinal absorption of zinc, yes, it could be done, but you need zinc 70, a stable isotope, to do this study. Not all laboratories have this methodology. And for chronic zinc status, I think assay of nail zinc by LIBS technique will prove to be a very good method. So this is my concluding slide. I think this is the end of it. Yes, I have many collaborators. I have only mentioned some of them over here. And I reviewed to you more or less 50 years of work. So I hope it made sense to you. Thank you very much.